You're listening to Medscape's In Discussion series on psoriasis, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Stephen R. Feldman, Professor of Dermatology, Pathology, and Social Sciences and Health Policy at Wake Forest University School of Medicine in Winston-Salem, North Carolina. Relevant disclosures can be found with the episode show notes on medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for you as healthcare professionals.
Welcome to Medscape In Discussion Psoriasis. I'm your host, Dr. Steve Feldman. This is the 5th episode of our 2nd season, and we'll be talking about biologics versus biosimilars. How do they compare? What are the major differences? And what do we know about patients' struggles and successes with them? To speak to these questions, we've invited Dr. Andy Blauvelt from Portland, Oregon, the new chair of the medical board of the National Psoriasis Foundation. He's also held senior staff positions at the NIH, Oregon Health and Science University, the Portland VA, and the Oregon Medical Research Center, a dedicated trial center where he was president for the last 10 years. Andy serves now as a key scientific advisor and teacher, sharing his vast knowledge on immunology, psoriasis, atopic dermatitis, and clinical trials. Welcome, Andy Blauvelt.
All right. Thank you, Steve. Happy to be here.
You know, Andy, my career compared to yours has been boring. I've been doing the same academic dermatology job for more than 30 years. You've reinvented yourself after successful careers in basic science, clinical research, administration. God forbid if I ever have to be an administrator. What's been the most rewarding thing for you?
Well, I actually didn't plan it this way, Steve. I mean, you know how life throws us curveballs. And so when I was at the NIH, I was there for a long time, and I kind of thought of myself as being a derm scientist. Never imagined that I would leave. I really loved it there. The NIH, as you know, special place, especially when Steve Katz was there. And the curveball is I met a woman, and you know her. My current wife, Molly, lived in Portland. And it was actually psoriasis that brought us together. She worked at the National Psoriasis Foundation. And so I moved out to Portland to be with her. And then I did the professor thing for a while. So I got a taste of what you do, which was also a very fun job. And I loved it. I loved the teaching. I loved the residents, kind of the mixture of everything. Started doing trials there, kind of a little bit of everything, which I really loved. But then the curveball was I didn't get along with my boss so great. And I just didn't feel, super comfortable there, even though I liked my colleagues and the residents. And so I fell into this opportunity to do trials, and it's really been great. So what I say is that not any one has been better than the other, that I have loved each one of my kind of separate things. And especially if you think about trials, right? The last 12 years or so, just been at the center of every biologic trial and every JAK inhibitor trial. And it's been a lot of fun the last 12 years doing just studies.
How lucky you were to have a boss that you didn't get along with.
Yeah. And I wanted to run things. I had that itch of being the leader. And I realized it wasn't going to happen for me at that university. So I bought this trial business. So I became the president of it and the owner. And so that satisfied my leadership itch or whatever. So
it lets you, as you say, be at the center of so much development. But even before this, when you were at the NIH, you were doing the basic immunology, if I understand things right, of what became these revolutionary treatments.
Well, I say that I've ridden 3 waves, and there's some luck in that, right? So in the 90s, I did AIDS research. And AIDS research was the thing to do in the 90s, right? So with Clinton coming on board, he said, I'm going to double the NIH budget. And he did. And we had this flurry of AIDS research, and a lot of it was done at the NIH. And I was at the center of it. I had a chance to work with Tony Fauci, one of the smartest men I ever met in my life. That was fun, interesting. And then when I moved to Portland, this new molecule called IL-23 had just been discovered. And I said, okay, I'm going to switch fields. The AIDS studies have kind of matured, and I'm going to switch and study IL-23 in psoriasis. And so I had a chance to kind of do the preclinical laboratory, focus on that cytokine in psoriasis pathogenesis. And as we know, the translation of that work went crazy, right? With IL-17 blockers, IL-23 blockers. And then I was at the center of that, the translational part of it. So I've kind of ridden these three waves and been lucky to do that.
That last wave, doing the clinical trial research on these drugs like IL-23 inhibitors, IL-17 blockers, before those came TNF inhibitors, and those drugs revolutionized our management of psoriasis. People would walk as I didn't know how sick I was until you put me on this TNF blocker. And now I don't prescribe TNF blockers because I got drugs that are safer and more effective.
Yeah, it really is a main message. And I think you and I are consistent with one another on that topic. It's one of my main messages when I give talks is that the 17 and 23 inhibitors are superior in terms of the three things we think about and care about, and that's efficacy, safety, and convenience. They are hitting on all cylinders. And so it is true. I'm the same way. I don't recommend using TNF blockers unless it's, let's say, PSA patients with psoriatic arthritis, and the 17 blockers are my drug of choice for PSA. But I'll use a TNF if their joints are not doing well with a 17 blocker.
Yeah. So we covered three of the four cylinders. The fourth cylinder is access and cost. And that's maybe where these innovator biologics let us down, and brings us to the topic of today's discussion, biosimilars, sort of the generic equivalent for biologics. Are they going to solve that fourth issue?
So there's a lot to talk about. So I think it's the promise of biosimilars is similar to the promise of any generic drugs. And that is, you have the same drug, maybe an awesome drug, it's been out for years, and now it's a fraction of the cost. And now everybody can use it. That's sort of the promise of generics, right? I'm just speaking about generics now, but it is all applicable to biosimilars because it's the same concept. When we see generics priced so high, it's like, oh my gosh, but it's generic. And that's frustrating to us because generics, we think, should be cheap because they're generic, by definition. And we kind of are thinking this way or hoping this way with biosimilars too, because they're essentially, as you said, biologic generics, and it just is not what's been happening in the U.S. So we see discounts of 10%, maybe 20%, and it's completely different in Europe, where they can negotiate the drug prices, and essentially it is a fraction of what it was. So friends of mine in Europe have said that it's now as little as $500 per year for adalimumab, which is $40,000, $50,000, $60,000 a year in this country. So it's been realized, sort of the promise in Europe, but here it's all about what the market can bear, right? That's the answer we always get. Oh, what the market can bear. That's why the drug prices are so high here, and a whole bunch of other reasons. And so it's disappointing to me. I think biosims in this country will be cheaper, yes, but they're not going to be so cheap that, oh gosh, now everybody can get a biologic.
Yeah. Sounds like insurers ought to be paying for medical vacations in Europe.
Yeah.
Okay. We've been talking like biosimilars are the same as generics, that they're the same medicine as the innovator. But I think our listeners may know that biologics are so large and such complicated glycoprotein with more than just sequence, but secondary, tertiary, quaternary structure that can vary, that it's so complicated you cannot duplicate a biologic. Let's talk about how biosimilars and biologics are similar and different.
It's a great question, and gets to how they're made. Majority of drugs are chemical compounds. I call them chemical compounds. They're made through chemistry experiments. You have a chemical reaction and you get the compound in your powder, like back in organic chemistry, right? You make a drug that way. Biologics are different. They're called biologics because they're made from living cells. And so what happens is you have a cell line that's growing in a culture supernatant, like in case of biologics, the big vats, these huge vats of liquid. And in the liquid are cells that are growing, live cells, and they've been transfected with DNA to produce a single antibody. And so the little cells are little factories for the biologics, and they're producing the same antibody because they've been transfected over and over and over and over again. And then what the manufacturers do is collect the soup and get rid of all the cell components, and they have the antibody left over. And it's a monoclonal antibody, so it's a single antibody that's being made by those cells. And so we call it originator biologic. So Humira, Enbrel, the names we know, Stelara, they have a single cell line, and that's proprietary. But what's not proprietary is the transfected material. So what biosimilar companies have done is get a similar cell line and use the same transfected material. And so their cell line is making a similar drug. And you're right that because it's an antibody, and antibodies are secreted from cell surfaces, they pick up lots of tertiary structures and carbohydrates and so forth. And so to characterize a biosimilar, they have extensive preclinical studies comparing side by side the biosim and the originator biologic, it's called. And they do functional assays. They look at the primary structure, secondary structure, tertiary structure, extensive preclinical work. And then the funny thing is, to get them approved, they only need one study, just a single study to get approval. And so the work, if you will, is in the preclinical work of the biosim and very little clinical work, whereas it's the opposite in originator biologic, where there is preclinical characterization, but you see these huge, clinical programs that cost, hundreds of millions of dollars to do a clinical program for a new biologic. But the clinical programs for biosims are very small. And that's why a lot of companies are doing them, because it is cheaper. It's like a tenth of the cost to develop and get a biosim FDA approved compared to an originator biologic. And the cost mainly comes, again, from clinical trial work. And that other crazy thing, Steve, about this is that for TNF blockers, you can do the one study in any indication, like rheumatoid arthritis, and then it's approved for all the indications: psoriasis, psoriatic arthritis, Crohn's disease. The biosim companies don't have to do studies in all those diseases. They just pick one disease, do one study, and they compare it to the originator. So it's a kind of a side-by-side. And if it behaves preclinically like the biologic in all those assays, and if it behaves similarly in the clinical study, whatever study they do, then the FDA says good to go.
So basically, you start with a beer factory and you grow up these cells, you purify the stuff, you have an identical amino acid structure, you show in preclinical studies that it binds the target basically identically.
Yes.
You show in people that the pharmacoeconomics, you inject it into people, the blood levels are basically identical. Assuming there's no magic in the world, the drug should work the same. And in top of that, you get a clinical trial showing that actually in humans, it does work. I don't even know if the clinical trial is required. Of course, they always do at least one. But what I find interesting is people are concerned that there may still be some difference between the biosimilar and the originator. And I think what they're missing is that the originator is so complicated. Even the originator company cannot duplicate their product from batch to batch. And so we actually have more information on the current batch of the biosimilar than we do on the current batch of the innovator. Hadn't gone through any clinical trial unless a biosimilar company puts it through a clinical trial. So you've got more data on the biosimilar than we do have on the current batch of the innovator.
So, Steve, that's really interesting, because years ago, I became the head of the Biosimilar Committee for the International Psoriasis Council. And at that time, maybe about 7 years ago or so, one of the big issues, are they going to be equally efficacious and equally safe? Because immunogenicity, as you know, can be an issue with any biologic therapy. You can develop antibodies to the drug, anti-drug antibodies. And so that was a big thing. It was like, okay, are these going to be more immunogenic? Are they going to be as effective? And we have tons of data now And I've been involved in review papers as well, looking at all the efficacy and all of the safety data, which includes immunogenicity. And most of it comes from Europe because the TNF biosimilars have been used in Europe for many years now. And it's the same. There are no issues that have arisen. And I've extensively, again, looked at the data and the primary studies, and they just don't look different. And so that's very good news because we did not know that, right? We didn't know that at the beginning. And that's coming from real world evidence because when they're out there and being used and in cohorts and so forth, that's where the rubber hits the road. We have a ton of real world evidence in Europe that essentially they're performing just like the originator. And that's good news for us.
Yeah. Well, I think of a biosimilar as kind of a lot like another batch of the innovator, but are there still challenges associated with switching patients from an innovator product to a biosimilar?
Yeah, that's been studied too. So the basic study is to start a patient on an originator or a biosim, and then you can look at efficacy, the primary endpoint, and then you can do a single switch. You can go from the originator to the biosim to the originator, or you can design a study where there's multiple switches. So we've done those at Oregon Medical Research Center where we know the patient's going back and forth between the originator and the biosim. And guess what? Nothing happens. Nothing happens. So we have real world evidence as well on single switches and on multiple switches. And it's essentially the same. So really for me, it boils down to cost.
Yeah. So how about educating patients and doctors about this? Anything we need to be doing?
Yeah, we haven't talked about this, but I'm going to take this back to what we talked about at the beginning, because the key thing for me, for dermatologists right now, is we just said it a few minutes ago, 23s and 17s are where it's at with psoriasis treatment. Superior efficacy, superior safety, superior convenience. And yet we don't have biosims for 17s and 23s. So those costs are going to continue to stay high for those drugs. So let's have a scenario now where the dermatologist wants to use a 23 or 17, and now the insurance company or the payer or whatever has gotten a deal on a biosim. And guess what? It's a TNF blocker. And so it's not so much a problem with the biosims. It's sort of the competitors are better type thing. So now where I think biosims will make the biggest impact is in rheumatology, right? Because in rheumatology, TNFs are still dominant. And so there they should take over. There shouldn't be any more originator use in rheum conditions because essentially have the same drugs that are cheaper. But in derm, I fear we're going to be forced, if you will, quote unquote, forced to use a TNF blocker because of cost when we really don't want to use that drug in our patient sitting in front of us. We'd rather use a better drug.
One of the IL-23 blocking drugs also blocks interleukin-12. It's the oldest of these interleukin-23 blocking drugs, ustekinumab. Man, that used to be one of my favorite treatments for patients. I understand it's going to be biosimilar quite soon.
That's a good question also. And thanks for bringing it up. And so what happened with the TNF? So I'm going to clarify because I said that TNF biosimilars have been out and about and used for many years in Europe, but just now being approved in the US. Why is that the case? So that is a legal thing. The US lawyers have been able to basically finagle the patents and extend the patents in the US, and the Europeans were having none of that. When the patent went up, the patent went up. They weren't extended in Europe. So legally, those originator companies in this country were able to keep it from coming out. And what's happened is ustekinumab, as you just mentioned, is not going through that same kind of legal thing. So now that's coming up. And it'll be, I think it's 2024. So it was approved in 2009. And I think it's a 15-year patent. And so, yeah, and we've been doing those studies, ustekinumab biosims, and guess what? They're the same as the originator. So we're getting the same result. So I think that will be better for dermatologists. Let's say if we do have that situation where we're forced to use a biosim, well, starting next year, I'd rather use ustekinumab than a TNF biosim.
Are you at all concerned that patients are going to be leery of biosimilars and they're going to ascribe any headache, nausea, dizziness that they happen to get at the same time that they switch as being caused by the switch to a biosimilar? And how might you manage that?
I think it's natural to what you just said, to if there's going to be any issues with a patient on a biosim, to maybe attribute it to the fact that it's a biosim, right, and not the originator. But the fact is, these drugs are made by the same companies that are making the originator drugs. And so, for example, just one example, right? Amgen is the makers of Enbrel. Amgen is a big biosim company, and on and on. Novartis, the makers of Cosentyx, are a big biosim company. So it's just two examples, but there are many. So in other words, they know how to do it. They know how to do the quality control. They know how to do the studies. They're really good at it. So I think it's natural to say, oh, it's not the originator. It may be something with that, that generic biosimilar type thing, that weird. They're really not weird. I'm trying to reassure your listeners that it's basically the same. And to patients, I say generic. I even though, technically between you and I, it's not generic. I think the patients understand the word generic and they're okay with it.
I like that. All right. Well, what's one thing you want our physician listeners to do differently after hearing this podcast?
Well, going back to our original thing, since I have a forum here, if you have a choice, you should be treating all your patients with a 17 or a 23 blocker. Incredible high efficacy, incredible safety margins. None of the many things we used to worry about with TNF blockers are occurring with those classes of drugs, like serious infections or cancers or heart issues or hepatitis B reactivation. They're just superior, both classes. And I use both and I recommend both. I do favor 17s with PSA patients and I favor 23s if there's no PSA. But I usually put the top two 23s and top two 17s and say, look, if you're using one of those four drugs, you're in good shape. That's my main message these days to dermatologists. And as far as biosims, maybe if your patient can't get on anything else, then yeah, go for biosim adalimumab. It's certainly better than topical therapy or methotrexate for somebody with moderate to severe disease.
Well, we've discussed how biologics have been revolutionary in the management of psoriasis. We have some that are just amazing, the IL-17 and the newest IL-23 blockers. We have biosimilars of some of our older biologics coming. They were revolutionary in their own way, maybe not as good as what we have today, but we may find insurers encouraging us to use them before they cover the newest agents. Andy, thank you so much for being on the program today. And I want to thank our listeners for joining our discussion with our guest, Dr. Andy Blauvelt. And be sure to look out for our final Season 2 episode with Dr. Megan Noe on the topic of pustular psoriasis. There's much more ahead, so be sure to check out the Medscape mobile app and share, save, and subscribe if you enjoyed this episode. I'm Dr. Steve Feldman for Medscape in Discussion.
Thanks for listening to Medscape's In Discussion Psoriasis podcast series with our host, Dr. Stephen R. Feldman. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out medscape.com or the Medscape app for show notes, links, and more information on psoriasis.